Dave mentioned, my name is Annabelle, and today I will be talking to you about creating the best brew, the role of mitochondrial membrane potential in assessing the myths of making beer. So, before we get too crazy, let's talk about what yeast are. So they are a model organism. You'll see a picture of them right here, as well as them in flesh form. I think they're very cute. They are eukaryotic organisms just like us, which means that even though they don't look anything like us, there are lots of similarities between yeast cells and humans. They are well characterized, which just means that we know a lot about their genomes. We know the differences between genomes of different species, and this information is pretty readily available. They have a short lifespan, which is great because I'm only planning to be here 5 years, and I don't have time to wait for them to grow up to see if my experiment worked. And they're cheap, which is great because I am broke and Lawrence doesn't like spending money nowadays. So what can we do with it? Like I mentioned before, we can use it for scientific research. So we can use yeast to study things like genetics, metabolism, cancer, Alzheimer's, literally anything you can think of we can study in some way, shape, or form with yeast. You can also make drugs with yeast. So a bunch of scientists have actually genetically engineered yeast to make drugs for them. So instead of all of you organic chemists out there slaving away over your reactions, I can just say, yeast minion, make me this drug, and go off and do whatever I want until we're done. But the most exciting thing that yeast can do for college students. So, my talk will be focusing on connecting scientific research with yeast making alcohol, and the specific type of alcohol I'll be talking about is beer. So, how to make beer? You start out with some grains. This is usually barley, but can be wheat. This can be rice, can be corn, can be literally any type of grain, but usually we use barley. And you add that to water, mash it all up, and during this mashing process, you're trying to extract sugars from these grains. You filter your mixture, and then your liquid is now called wort, and this liquid is now full of all the sugars that you pulled out of your grains. Next, you boil it to get rid of bacteria and other impurities, and then you also add hops and continue boiling to get more bitter flavors out of these hops. Once that has cooled down, we add yeast. And this is the step that I'm most concerned about, is this fermentation step right here where you add yeast to your sugar water, and then your yeast get busy eating up all those sugars and making wonderful alcohol and carbon dioxide for us. So once they're done with that, it's bottled, you can drink it. Hooray! So how do we study brewing? We use a process called translational brewing, which basically means that we translate scientific research into use for the industry. So we have our lab on the left, and we have our brewery on the right, and they're kind of two separate entities. And translational brewing is basically the bridge that connects them. So what we did with this is try to replicate brewery conditions in a laboratory so that we can apply things that we learn in the lab to real-life situations. So what can we look at using translational brewing? We can look at some of the myths of brewing, such as brewing in copper kettles, which is thought to make yeast grow better. We can also look at something called stuck fermentation. So fermentation is the step in which you have all your sugar water coming out, and you got your little Pac-Man yeast going on, eating it, and then they make carbon dioxide and alcohol. So this is a normal fermentation. In a stuck fermentation, it basically is stuck. Your yeast doesn't eat all the sugars, and your fermentation is stuck, hence why it's called a stuck fermentation. So brewing right now is not much of a science. A lot of what brewers base their decisions on is based on things like smelling, tasting, and observing. So if your beer tastes really good, tastes really bad. If it smells kind of funny. If it looks green, probably not good. But what we're concerned with is, can we find a quantitative measurement to study fermentation? Turns out there is such a thing, and it's called the mitochondrial membrane potential, or MMP. So what does all that mean? So this is the mitochondria. You ever taken BuzzFeed quiz? It is the powerhouse of the cell, responsible for making most of the energy that a cell uses. So as you can see, you've got a brown-colored outer membrane and a tan-colored inner membrane. So there are two membranes in this organelle. You also have a blue-colored matrix, and this is just a space inside the inner membrane. And then you have a black part between the membranes that is called the intermembrane space. And floating around in this black intermembrane space and the matrix are a bunch of protons. So as the mitochondria is producing energy, it is pumping protons across this tan inner membrane, and that creates a difference in proton concentration between the intermembrane space and the matrix. And that difference is what we call the mitochondrial membrane potential. Now, as your mitochondria is making energy, it converts ADP to ATP. And ATP, if you take the BuzzFeed quizzes, is the energy currency of the cell. So while it does that, it also turns NADH into NAD+. And so these two energy-rich compounds are going to be important later when I talk about flavor. So what can we study using this? We can study things like metabolism, because as I mentioned earlier, it is the powerhouse of the cell, so directly related to metabolism. And that can be tied into aging as well, because as we get older, our metabolisms get worse. I'm not looking forward to that. We can also look at things like flavor and viability, and this has to do with the NAD+, NADH ratio, which if you remember from before, as your mitochondria makes ATP, it turns NADH into NAD+. And that ratio affects the flavor compounds. So if you have a high mitochondrial membrane potential, that means you have a low concentration of protons floating around in the intramembrane space. That means more of it is bound to NAD+, which means you have more NADH, and that means less flavor compounds. So we have our mitochondrial membrane potential. We have a way to measure fermentation, but how do we measure mitochondrial membrane potential? So it turns out we use this magical instrument called the flow cytometer. So what this basically does is you load your sample up here, and it runs it through one cell at a time. And as they're running through, the flow cytometer hits them with a laser, and if it happens to hit a cell that we've tagged with a fluorophore, it will cause that cell to fluoresce, and then it will go to a detector, and the computer will show me an image kind of like this. So here we have forward scatter, which just tells us the size of our colonies, and then here we have side scatter, which tells us how complex our colonies are. And each one of these dots represents one individual yeast cell that went through. So the computer will take all this and then combine it into a number that I can then use for studying. So how do we actually measure mitochondrial membrane potential? Because forward side, like knowing the size and complexity of your cells is important, but like we want to specifically look at mitochondrial membrane potential. So to do that, we use fluorophores. We use 2 of them in my research. One of them is called propidium iodide. Looks like that. It intercalates DNA. So what that means is it gets in between the base pairs of DNA and binds to that. And this allows me to only look at living cells. I can screen them off. And then the second one that I use is called rhodamine 123. And this binds to protons and defluoresces. So this can tell us about mitochondrial membrane potential because it is the difference in proton concentration between the intramembrane space and the matrix. So a high fluorescence means a low mitochondrial membrane potential because there are fewer protons around, so it will not defluoresce as often. This is kind of counterintuitive. So back to translational brewing. We have our quantitative measurement. Can we replicate large-scale brewing conditions in the lab? Turns out we can, thanks to a former student called Greg, named Greg. His senior experience was entitled In the Quest for a Better Beer, the Role of Mitochondria in Industrial Brewing. And so what he did every day was go across the river to Stone Arch Brew Pub and study large-scale brewing there. So he went over there every day and took samples from their giant fermenters and came back and analyzed them in the lab. And what he did later was replicate his experiment in the lab here. And the solution that we came up with is this pico brewery. So as you can see, it is significantly smaller than those giant fermenters. It is a 100 milliliter round bottom flask. It contains 75 milliliters of wort or YPD, and that is just liquid media that contains lots of sugars for the yeast to eat up. And it also contains 1 milliliter of yeast. So as you can see, these are airlocked, so you have this glass stopper here that's hollow, and then you have this hollow tube that goes all the way around into a test tube filled with water. And we did that because as yeast chew up sugars, they create alcohol, but they also create carbon dioxide. So we want a way for carbon dioxide to get out, because we don't want any exploding glass. And we also want a way to keep air from getting in. We don't want bacteria getting in there. We don't want any contaminants. So this setup allows the gas to escape but nothing to get in. And then every day we took samples from them. So what can we look at using a pico brewer? We can look at how different types of brewing yeast or wild yeast metabolize different types of sugars. We can look at the effects of various metals on fermentation. And we can also look at the effects of viruses on stuck fermentation. So what do we actually do with our samples? We have our pico brewery here. This is day 1. And like I said earlier, take a sample from it every day. So day 1, day 2, day 3, we take daily samples. And so the first 3 days are what we classify as generation 1. And on the 3rd day, we take yeast from this old pico brewer. So this is the same pico brewer, just different days. And you take yeast on the bottom of here, and then pitch it into a fresh pico brewer with lots of fresh media for it. And then on the 4th day, we take another sample, more daily samples. And this we call generation 2. So this repitching of yeast is done to imitate what they do in a brewery setting, where every couple days they will take yeast from their old media and give them fresh media to chew up. So what do we do with the samples after we took them? We treated them with propydium iodide, which once again allows me to only look at living cells. We also treated them with rhodamine 123, which allows us to look at mitochondrial membrane potential, and then we ran them through the flow cytometer. Everybody with me? So what do we actually look at? Turns out there are so many things that we can look at. You know what that means? Dave is super excited. So Greg looked at changes in mitochondrial membrane potential after generations of reuse. So like I mentioned earlier, brewers will repitch yeast for generations. And what Greg did was look at the differences in mitochondrial membrane potential after years, not years, days and days of reusing them. So we actually looked at a lot of different things, but we don't have time to talk about them. So I'm going to focus on 2 of the things that we looked at. So the first one is the effects of copper on fermentation, specifically asking the question, does it improve mitochondrial membrane potential? And we're going to look at stuck fermentation, asking the question, why does this happen? So let's get started. Let's talk about copper. So all organisms need small amounts of various metals throughout their daily lives, and this helps them with biochemical reactions. Now, the Germans have been brewing in copper pots for hundreds of years. And German beer is great, so there must be, must be right somehow. So they brew in pots that look like this, and they believe that this improves fermentation. And we know that's true because German beer tastes really good, so something must be going right there. However, there is no biochemical basis for this, so we decided to jump in and see if we can figure it out. So our experiment. 2 pico breweries, and we put yeast into each one of them. One of them we only have yeast, and that is our control. And in the second one we add 0.04 millimolar of copper chloride, and that is our experimental beer. So after running them for 15 days, this is what we got. So this is a British ale yeast, and we have time in days on the x-axis and geometric mean on the y-axis. And that's just the average of the 30,000 cells that I ran through the flow cytometer for each sample every day. So each one of these dots is an average of 30,000 samples. So as you can see, we have a control in blue and copper in red, or experimental in red. So the first couple days is kind of a mess, which is what we expect, because if you get sneezed on by someone who's sick, you don't instantly start sneezing and getting super sick. Your body takes time to process whatever it's been infected with before displaying symptoms. So same thing happens with yeast, where it takes time for them to get used to their new environment that now has copper in it. And once it's gotten used to it, you see there's a really big difference between the experimental and the control groups, where the experimental group has a consistently lower level of fluorescence, which means that it has a higher mitochondrial membrane potential. So these lines never cross, which means that it stays consistently higher. So from this, we can conclude that copper improves fermentation by increasing mitochondrial membrane potential. And we did this with multiple different types of yeast, but I'm only showing one example. So that's all well and grand. It means Dave is really happy. But being data horror that he is, he was not satisfied. Said, I want more data, young grasshopper. So what can you do when your research advisor tells you he wants more data? So, moving on to the next project. We looked at stuck fermentation. So, to remind you all of what that is, fermentation is a step where you have all these sugars hanging out in your wort or YPD. You add yeast, mix it up really happily, makes carbon dioxide and alcohol. So in a normal fermentation, so in a stuck fermentation, it starts out as normal, where your yeast is happily chewing away, doing what it's supposed to do. But then in a stuck fermentation, it stops. The yeast don't eat up all the sugars completely, and nobody really knows why this happens. So brewers want to avoid stuck fermentation because it can cause bad flavor, which we don't like. It's also kind of a waste of yeast and wort, and the way they fix it is by adding oxygen to kind of kick it off. Because if you remember, most of this fermentation is done without any access to fresh oxygen, so adding oxygen could help kick it off. Or we can add more yeast in there, and hopefully by adding new buddies in there, they will be encouraged to keep eating. Our hypothesis is that killer yeast viruses cause stuck fermentation. So how do they do that? Has no one seen this movie? Okay. Okay, so what are killer yeast viruses? They're the only virus known to infect yeast. They are a member of the totivirus family, and they are double-stranded RNA viruses. So your infected yeast, once the virus has gotten in there and gotten it sick and everything, it causes your yeast to make toxins and secrete them into the environment, and that then kills the surrounding yeast. So then we call our infected yeast the killer yeast, and luckily for them, they are immune to the toxins that they secrete, but the unfortunate yeast surrounding them is going to die. And as you can expect, this can spoil brewing. So how do we study this? We went through and isolated wild yeast. So the first time we started this project, we used domesticated yeast, didn't find any viruses whatsoever. So we figured maybe domesticated yeast have been so inbred that there aren't very many viruses left. So we decided to look at wild yeast, because if anything's happening, it's going to be with the wild yeast. Because they're all hanging out in the wild and being happy. So we did wild yeast isolation, and then ran virus picobreweries using the viral DNA samples that we pulled out from the wild yeast. And we asked questions, are brewing yeast infectable by killer viruses? And this is really important for anyone who's working with domesticated organisms, because if you keep using the same organism, for example, like orange trees in Florida are dying right now from a virus because after years and years of only using the same type of tree, once you have a virus that gets in there that can destroy one tree, you can destroy everything. So we don't want that with beer, because we want to keep drinking beer for the rest of our lives. Second question, will virus particles affect mitochondrial membrane potential? Our hypothesis for kind of why stuck fermentation happens is that you have your normal fermenter hanging out, and the virus just falls in there on accident. It replicates, and then gets your fermentation stuck. So we're going to test that. We have 6 wild yeast samples. I know there are 5 stars, I can count. So this first sample is from northern Michigan, and all of these samples were collected from the wide travels of Dr. Dave. This is from northern Michigan, from a bunch of wildflowers. The sample here is from the Tennessee-Kentucky border, from an oak tree. We have 2 samples from Costa Rica, one from a basque tree, which orchids like to grow on, and we all know Dr. David loves orchids. And the second one from Costa Rica is from Poaz, which is a volcano. That's super cool. And then up here we have a sample from Norway, up in the mountains. So from some small shrubs, no trees this time, because they don't grow at that altitude. And then we have a sample from an olive tree in Sicily. So that's good. What do we do with them? We isolated virus DNA and RNA. So we grew our samples out, centrifuged them really, really hard, filter sterilized them, and then we then had 6 individual samples of each one. We also had a viral mix which contains some of all 6 of our samples. So what we did next is kind of what we did before. So we had the 2 picobreweries, and in each one we add yeast, and one of them is controlled because it just has yeast. And in one of them we add our viral mix, and that's the experimental group. So we used the viral group and not the individual samples because no one's really done this before, and we weren't sure what was going to happen. We don't know if there's anything in there at all, and we don't know if that will affect the yeast in any way. So we're like, this has all 6 of them. If they're going to do something, this is the one that is most likely to have an effect. So we did this with 4 different brewing yeasts, which means we have 8 picobreweries, 4 controlled, and 4 that have viral mix in it. So what kind of brewing yeast did we use? They can be divided into 2 types, so top fermenting and bottom fermenting. And what this basically means is that in a top fermenting yeast, such as an ale, your yeasts are hanging up here in the top of the fermenter. And in a bottom fermenting one, like a lager, they're hanging up down here. Simple enough. So we have 3 different types of top fermenting yeast. One is a British ale yeast. we have a Belgian Saison yeast, and a Dusseldorf alt yeast, which is a German yeast. And for bottom fermenting, we have a German lager yeast. So all 4 of these yeasts are known for their distinctive flavor, as well as the way they're brewed. So if mitochondrial membrane potential is actually important in flavor, or actually affects flavor, we should see differences in mitochondrial membrane potential between these 4 yeasts. So after running those picobrews, we got another graph. So we found that viral infection does change mitochondrial membrane potential. So this is our German lager virus trial. So this is the bottom fermenting yeast from Germany. We have, once again, days down here and geometric mean on the y-axis. And we have control in blue and our virus sample in red. And as you can see, they start out very similarly on the first 2 days, like they're almost the same. And then on the 3rd day, something really weird happens, where both of them have a drop in fluorescence, which is an increase in mitochondrial membrane potential. But our virus sample has a higher level of fluorescence than our control, which means that it has a worse mitochondrial membrane potential than our control. And then on the 4th and 5th days, it goes back to being pretty much the same. And we see a similar thing with our British ale yeast. So this is a top fermenting yeast from a different country. So they should do different things, but they do very similar things. So here, once again, you can see that day 1 and day 2 are very similar, and then there's a giant spike on the 3rd day, where here you can really see that giant spike in fluorescence means a big drop in mitochondrial membrane potential. And then they go back to being really similar after that. So what can we conclude from this? Viral infection does affect mitochondrial membrane potential, where in both of the samples that we looked at, they started out with similar mitochondrial membrane potential, and there was a drop on the 3rd day, and then returned to very similar values after that. So our next step, which sample is responsible for this? So we chose to look at 005, which is our British ale yeast, because it seemed to be the most sensitive to viral infection. And we ran picobreweries using individual viral samples this time, instead of the big mix. Here are our results. Kind of looks like a mess, but I'm going to walk you through it. So we have our control in red right here. Hang on. And as you can see, our orchid basque from Costa Rica, from that tree in Costa Rica, does kind of what we saw earlier, where there's kind of a similar value in the first 2 days and then a big peak in fluorescence and then return to normal. So in this case, this sample affected mitochondrial membrane potential by decreasing it, because peak in fluorescence, decrease in mitochondrial membrane potential. Our Alpine Norway sample didn't really do much, just kind of hanging out. And then in the case of the Olive Sicily and the POAS, which is a volcano sample from Costa Rica, they both decreased in fluorescence, which means that it increased mitochondrial membrane potential. So what can we conclude from all this? Individual samples have different effects on mitochondrial membrane potential. So some of them increase it, some of them decrease it, some of them don't do anything. So our nice viral mix results are probably a result of a combination of different samples affecting it differently. So our next step could be to go back and figure out which exact samples are responsible for that beautiful curve that we saw. We also wanted to look at what exactly are these samples? What's affecting them? And what we did to study that is this killer yeast assay. And as of yesterday, this is what it looks like. So this is a work in progress. So what I did is I plated out a lot of yeast. This is the German lager yeast, and then went over and spotted in each of these circles either the virus mix or one of my individual virus samples. And as you can see right there, that is exactly what we want to see. I don't know why the duplicate didn't work. But if you remember from earlier in my talk, once a yeast has been infected, it is going to start secreting poisonous toxins that will then kill all the surrounding yeast. So if we spotted this with the virus and it has infected the yeast, it should start secreting toxins and nothing else should grow in that circle, which is exactly what we see right here. I really don't know why none of these grew. I'm going to have to retry this and replate this, and I don't know why my duplicate didn't grow. We also can't directly translate these results back to the graph because this is a different yeast. I tried to go the other one out and it didn't grow, so I'm going to try that one again. So, conclusions. Mitochondrial membrane potential is a quantitative way to study brewing. The myth of making beer in copper pots does have a biochemical basis, and stuck fermentation may well be caused by viruses. Future work, we can use mitochondrial membrane potential to monitor the brewing process. So look at different types of wort that have different types of sugar. Look at different types of yeast and how they metabolize these different types of sugar. Look at different temperatures, see how that affects mitochondrial membrane potential. We could also look at different concentrations of copper. We can look at different types of metals and their effects. We can also identify new brewing yeast. So one of the other projects that we did that I'm not talking about today is a massive wild yeast isolation project. And we can use mitochondrial membrane potential to maybe help find new types of brewing yeast that have different types of flavor that we then get to enjoy. And then finally, I want to identify the components of the wild yeast that are affecting mitochondrial membrane potential. Are they viruses? Are they bacteria? I don't really know. So keep running that killer yeast assay, see if I can get it to work. And then also sequence, so that we can then match up these sequences with the known sequences of killer yeast viruses that people have already studied. Acknowledgements? Gotta acknowledge Dave for taking me in and for putting up with me for all these years and sending me out, always being a really positive force in my life, except when he's making fun of me. I want to thank Eric Lillen upstairs for being available to answer any questions that Dave and I have had. I also want to thank Daniel for putting up with me all these years too. All the things that I want to use, all the YPD I've asked you to make, and all the sheaf fluid I've asked you to order. I also want to thank Greg and Stone Arch Brewery for all their preliminary research, and also my lab group. We have all here. References.
You noticed that there was a change in the MMT on day 3 specifically of all those different mixed yeasts.
Yeah.
Did that have anything to do with the fact that you were placing it in new media on those days? Was there also
for the virus trial we didn't do any new generations. We just like kind of dumped it in to see if it would do anything. I think in the future we might do some where we do repitch just to see if that affects it. But for this first pilot experiment, we're like, we don't know if anything's going to happen, so we're just kind of going to dump it in there.
So there were no regenerations
Yeah.
in that case? Okay.
Alex?
So I think it was when you were talking about rhodamine, you used the term defluoresce. Can you explain what that means?
It's gonna light up if it's not bound. So once it's bound to a proton, there's not gonna be any light. Does that make sense? So like if it's not bound and there are fewer protons, that means you have a lot of light glowing, and that means a lower mitochondrial membrane potential. But if it is bound, then you're not gonna see any light. If that makes any sense. Sarah?
Right, going back to this question,
Yeah.
why do you think the MMP went back to being similar on day 4? Like if it's stuck, I would expect it to kind of stay different, and so I don't really understand why it went back. And I was wondering, do you have any insight?
I don't really know. We haven't really looked at, gone into any detail with that, because we can't confirm one that it was stuck to begin with on that 3rd day, and that it got magically unstuck. I guess maybe thinking in the future, looking specifically into if it is actually stuck or not, and 2 of if it's actually a virus. We don't really know what's happening on that 3rd day, just that there is a difference. So that could be one of the things that we look at in the future, is to figure out what exactly is going on there. David?
For the virus versus the copper graphs, the virus is running for 5 days.
Copper?
That was 15. Was there a reason why?
Well, okay, the copper one we did 2 summers ago when we had bolt loads of time. The virus one I did 7 weeks ago, so I was just can I get this out?
Fair enough.
Brianna?
Do you know if there's any trends in what styles of beer tend to get stuck more? Because so you said that you don't think you could isolate any viruses from the domesticated beer. So would you say that maybe beers that include wild yeast, like maybe some saisons, would get stuck more if the viruses are? I mean, I know
there's been some research done on killer yeast viruses, but not really anything done on connecting that to brewing. So we're trying to figure all that out and see if we can maybe start our own thing. Because we could test in the future because we have all those wild yeast samples, we might as well go through them, and it has our wild yeast samples susceptible to these virus things that we put in there. Do we see the same effects? And then do maybe the hybrids that you made, we could test those too to see if having part of wild yeast DNA makes it more susceptible or more immune. I don't know.
Yeah.
Yeah.
So I have a few. I'm a little bit confused confused about the link you gave between the NAD plus and then the flavor compounds. And can you just go over that? You lost me there, and I'm just like a little bit more understanding of how that creates flavor compounds that would be outside.
Okay, so as your cell turns ADP into ATP, it turns NAD plus into NADH, which if you have more NAD plus, it will go through and turn all your esters into alcohols, which don't have as much flavor. So you want to have more NADH hanging around. I did that backwards. More NADH, less flavor. So more NADH means that more of your esters, which are basically your flavor compounds, right? And we want more of those.
So you're saying with NADH, you're going to get more esters?
More alcohols.
More alcohols?
Yeah.
Okay. Okay.
Does that make sense?
Yes, it does. Thank you. So how did you decide on what copper concentration to spike your copper kettle with? Because that just seems super high to me from what you get off of copper kettle. And so I'm just curious how you decided on that.
So we got it from two different places. One is from a paper by Dan Thorson, who went through and we literally just went through his methods and he says use 0.04. But there's also this great paper from 1908.
I was like, I knew I was going to ask
Someone is going to ask this question. So
You're right.
Yeah, based off of this paper and Dan's work, we decided to use that. We haven't tried other concentrations. We might do that in the future just to see if that is the right concentration to use. Yeah, this is what we based up our decision off of.
So this paper itself went and measured how much copper was in?
Back in the day, they were like, use a grain of copper.
Okay, and then is it a copper, I'm sorry, I just have lots of questions. So then is it a copper salt you're putting in? And then what
we
copper
use
salt?
Yeah, we use the salt.
What was it?
Copper chloride.
Thank you. Because that's going to produce the chloride in there and cause a whole lot of trouble. Okay.
But we didn't drink any of it, so we're all like,
I didn't realize anyway, it just tastes like it.
there really isn't very much research done on copper and use in brewing, so we were just trying to find some sort of preliminary research to base it off of.
I was just looking for logic.
Yeah.
Thank you. Then I just have a question about your repitching. So are you doing any quality control as you do the repitching to decide what parts to take? Because breweries do a lot of quality control with repitching.
What we did was we would just take a milliliter of yeast from the bottom,
Okay.
and then repitch it, and then keep the rest of that yeast and freeze it in case we wanted to go back and look
But
at it.
no checking to see if there's other contaminants that have got in there in the meantime.
I mean, we looked at it and it didn't seem to be contaminated, so we were just.
Okay, I guess I have So I know you said your pico breweries are very easy to scale up. I know that breweries themselves have a harder time just scaling up from their pilot systems. Can you give me insight of what's the difference in the pico breweries versus the scaling up, or is this just that the scientific questions are the same rather than the actual flavor equaling the same?
We